Next talk by Tanaya, Bose, and Ada Yonat from Weizmann. And the title is Origin of Life, Peptide Bond Forms by Protoribosome Constructs Links to the RNA world, from the RNA worlds to the RNA protein lives. Please.
So, good morning, everyone. I am Tanaya from the group of Professor Adayona. Our group working on ribosomes were interested in looking into how was the first peptide bond formed on Earth, or what is the smallest unit of ribosome in the prebiotic world which could form a peptide bond. So, thank you for joining me for my talk entitled Glimpse into Origin of Life from RNA to Protein. Ribosome, the protein synthesis organelle, comprises primarily of two subunits, the small subunit and the large subunit. The small subunit or 30S in bacteria provides the decoding site while the large one is responsible for the peptide bond formation. This peptide bond formation center is composed solely of ribosomal RNA and is known as the peptidyl transferase center or PTC. The PTC, situated in the heart of the contemporary ribosome, is composed of a semi-symmetrical pocket-like RNA fold. This figure here demonstrates the symmetrical pocket in the ribosome. A closer look into this figure, as well as the two-dimensional view of the symmetrical region in the peptidyl transferase center, clearly demonstrates the semi-symmetrical nature. Structural analysis and overlay of the ribosome complex of various species, which has been listed here, indicates that this region is highly conserved among many species of life. This PTC comprises of A region and the P region, which has been marked in blue and green respectively here. In addition, structural analysis of the ribosome with tRNA as well as their shorter analogues, which mimic the aminoacylated 3' termini bound to the A site and P site of the termini, indicates that the CCA 3' end of PTC bound tRNAs are also related by a semi-twofold symmetry. Based on this observation, we propose that this pocket is still implanted in the contemporary ribosome, which in prebiotic world was useful for various RNA processes such as replication, cleavage, ligation, splicing, and phosphorylation. We hypothesize that the protoribosome made exclusively of RNA molecule was later probably snatched by various activated amino acids to form the peptide bond. So the protoribosome provides the scaffold for peptide bond formation by anchoring the 3' end of amino acid and peptidyl tRNA molecule. This elaborate architecture of the pocket provides the required framework for catalytic activity of the contemporary ribosome by positioning the substrate in a spatial orientation, optimal for peptide bond formation. On the basis of this observation, we synthesized 13 RNA constructs, while some of them mimic the A region of the PTC and some others mimic the P region. There were some sequences designed by us which mimic the entire pocket, that is the entire A and the P region of the protoribosome. On the basis of this, so after the synthesis of all of the RNA constructs, they were tested for their ability to dimerize, or I would rather say their ability to form this pocket, which would catalyze the peptide bond reaction. So for that, we annealed all of the RNAs with buffer, which comprised of magnesium chloride. The samples were then run on native polyacrylamide gel. So out of the 13 constructs that we had designed and prepared in the lab, 7 marked here in stars, they could form dimers. But an important point to note here is TTA1P1, which comprised of the entire pocket, was also able to form a dimer. Next, we screen all of these 13 sequences for the ability to form peptide bond. For this, we chose this reaction, which has been reported in literature, and it is known, this sequence, which does not utilize the entire tRNA, but only the terminal, the CCA terminal of or the conserved terminal of the tRNA, is known to work on 50S and give this product. And so for the P site substrate, CCA-PCB is the P site substrate and C-poromycin acts as the A site substrate here. The difference between C-poromycin and the original tRNA is it has an amide bond here rather than the ester bond which is present in the tRNA. So what does this amide bond do? This stops further reaction, and which makes it easier for us to evaluate the or analyze the samples. All the reactions of with all the 13 substrates, that all the 13 RNA constructs that were designed were performed at 37 degree for 24 hours. Next, after the reaction, we analyzed all of the samples using MALDI-TOF. So for 50S, which was our positive control, we see a product at the according to the calculated mass, but we do not see any starting material. That is, all of the starting material was consumed in the process. However, for EFP1C, which is one of the constructs that we had designed, a significant amount of starting material always remained. For our experiments, we used a negative control in which there was no RNA or ribosome, and CHCA, which is the matrix which serves as the background for the reaction. So out of the 13 constructs that we had designed, 5 of them, which includes one entire pocket like the TTA1P1, were able to form the peptide bond, or catalyze the peptide bond rather. So for to further validate that this the peptide bond formed is indeed a product, so we treated our after the reaction mixture was treated with zero point 0.5 molar NaOH followed by neutralization with acetic acid. So what will this treatment do? This treatment will of course chop off this phosphodiester bond here, but the amide bond should remain unaffected with the base treatment. Therefore, if we later check the sample on MALDI and find the rest of the sample, rest of the product intact, this should confirm that what we achieved at the end of the peptide reaction was indeed a peptide bond. So this is before the base treatment. A peak corresponding to the product was observed at 1262, but after base treatment, a hydrolyzed product, as we had assumed, was observed. So this clearly confirmed that the product that we have obtained is not just an artifact, it is rather a peptide bond formed. Later, after this, we wanted to explore the reaction at different temperatures. So we monitored our reaction at different temperatures of 15 degree, 25 degree, and 50 degree, along with 37 degree, which was used earlier. So we found that 50S worked under all conditions of temperature, and our other constructs, or the other RNA constructs that we had designed, TTP1C and TTP1 and AP1, like 3 of them were able to form peptide bond under all conditions, whereas 2 of them did not form peptide bond at lower or at elevated temperatures. Next, we wanted to look into the role of time in, because as you see that the product formed is quite less in comparison to the starting material that left behind. So we wanted to understand the role of time on the reaction. So we lowered our reaction time from 30 minutes, and we saw that at 30 minutes, even 50S was not able to form the product. Later on, increasing the time to 1 hour, 50S was able to form the product which was detectable on MALDI, but not our RNA construct. Later, at all other reaction times, like the higher reaction times, every time they could form a product. But a point to note here is, from 2 hours to 24 hours, there was no drastic change in the amount of product formed, neither was the starting material completely consumed. But there is a point which I would like to mention here, that MALDI-TOF is a qualitative technique and not a quantitative one. Next, we attempted to work on the concentration dependent, like find out the role of concentration on the peptide activity assay. So for this, we increased the concentration of the RNA constructs to about 35 times, that is from 2.8 micromolar, which was used earlier, to 100 micromolar. So either increasing the concentration to 35 times of the RNA construct did not drastically change the amount of the product formed, nor on varying the substrates. So after the experiment, after this activity assay, we wanted to find a correlation between the peptide activity assay and dimerization. So on the left is the list of the sequences which dimerizes, and on the right are the sequences or the constructs which could form the peptide bond. A a careful look at this shows that the sequences which forms peptide bonds were all forming dimers. This supports our hypothesis that only the constructs that dimerized, which mimically potentially mimics the pocket structure, could mediate the peptide bond formation. Now, to further understand the role of dimerization in uh, peptide activity, and so what we did is we tried to understand the role of salts. So, in absence of salts, potassium chloride and magnesium chloride, which were used in reaction mixtures earlier, so in absence of all of the salts, we saw that they do not form dimers, or in presence of potassium chloride, excess of potassium chloride, and but in absence of magnesium chloride, such that the excess potassium chloride suffices the magnesium chloride, loss of magnesium chloride, but in either of the conditions we do not see any dimerization. And also, in absence of salts or in absence of magnesium chloride but in presence of potassium chloride, we do not see peptide bond formation in any of the constructs that we had that were earlier forming peptide bond. This further confirms that a formation of a dimer or formation of the pocket is necessary for peptide bond formation. A summary of the entire work: a series of 13 constructs were prepared mimicking the A and P region of the PTC. 7 construct formed dimers in presence of salts. 5 of them successfully were able to catalyze peptide bond formation. Investigation of the reaction conditions were also discussed. Our results provide for the first time the experimental evidence for the formation of peptide bond by a conserved RNA entity that is still embedded at the contemporary ribosome of all organisms. The proto-ribosome is the missing link from RNA dominated to the RNA and protein dominated world and may mark the origin of life as we know them today. At the end of my presentation, I would like to thank Dr. Anand Bashan, Dr. Gil Fritkin, and Nikita Dinger, who were actively who actively participated in the experiments. Dr. Ilana Agman and Professor Ken Davidovich, who were the ex-members of the group who began this work and continued this work. And at the end, I would like to thank Professor Ada Yonath, who had faith in me and handed over this project to me so that I could pursue the work. And thank you all for your attention.
Thank you very much, Tanaya. That was really interesting. We have time for questions, please.
So we have one question, that is, how tolerant is the fragment reaction to the presence of plausible prebiotic impurities such as polyamines or other such molecules?
In presence of polyamines, we have really not tested them, but this could really be interesting to test them. Like, we have not tested the fragment reaction in presence of other polyamines.
And other molecules, not specifically polyamines.
Other molecules, not specifically polyamines, also we haven't tested. The reactions were all done in presence of these two substrates mainly.
Mm-hmm. And another question is, how tolerant are the fragments to mutations of different kinds?
Mutations to different kinds. Actually, it is very strange that even with slightest mutation in the sequence, for example, we also had a construct in which, like, all of the constructs were basically, major of them were from thermophilus, while we had one construct from E. coli, so which you could call a mutation from thermophilus, because it's just a difference of few bases. In spite of just a small difference, it neither formed a dimer nor did it form a peptide bond. So I guess slight mutations does make a difference. We are still working on it, but small mutations does make a difference and does not form peptide bond.
Mm-hmm. Okay, and we have one last question here. Is the mechanism of how the RNA dimers catalyze the peptide bond formation known? Do we know the mechanism?
No, we do not. We are still working on the mechanism. As you see, so Actually, in contemporary ribosomes, so what happens is the A and the P region, it brings the stereochemistry of the tRNA in a position suitable to form the peptide bond. But for the protoribosomes, we are actually working on it. As you have seen that all of the cases, our bottleneck is like really little, a small amount of product formation, which is also like a point, because in a prebiotic world, all of the products that were formed should ideally be of very low, like it cannot be a huge amount of product, or we cannot compare them with the contemporary ribosome now. So we are still working on it, like to work on the mechanism, or how the peptide bond, how the protoribosome helped it or catalyzed the peptide bond formation.
Hmm.
So, assuming that indeed you have identified the protoribosome correctly, and if we compare it to the current ribosome, the distance is very large. So, can you speculate on what could have been the next step from the proto to the current ribosome? And in particular, what qualities did it acquire the ribosome? Was it better speed of translation, higher accuracy? Any speculations?
What we assume is the smallest unit which form the peptide bond. So these proteins, because as we know, the contemporary ribosome also has some proteins in them. So these proteins which were formed during the process, rather formed like participated in the assembly of peptide bond formation, rather participated in the assembly of the ribosome, and which later made it more efficient towards peptide bond formation. And also, like, it also then moved to other functions of the ribosome, for example, the recognition.
Thank you very much.